Well, welcome everyone. I'm Linda Isaacson, the communications lead for EMSL, the Environmental Molecular Sciences Laboratory. The focus of today's webinar is to discuss opportunities for accelerating your research using the BioSANS, or the Biological Small Angle Neutron Scattering Instrument, through this FICUS call for proposals between EMSL, the Joint Genome Institute, and the Center for Structural Molecular Biology. Joining us today is Scott Lee, a molecular bioimaging expert, here to discuss the structural biology capabilities available at EMSL. Hugh O'Neill, the director of CSMB, here to discuss CSMB's capabilities. Himadri Prakasi, a professor at Washington University in St. Louis, here to discuss how he uses BioSANS to further his research in bioenergy production through cyanobacteria. And we'll hear from Marcus Foston, who's also a professor at Washington University, here to discuss how he uses BioSANS to further his research in novel ways to use lignocellulosic biomass. Now, after we hear from our presenters, we have plenty of time to answer all of your burning questions about this proposal call. Now, for those of you who may be new to the user facility proposal process, here's a quick primer to bring you up to speed. Now, user facilities like EMSL and JGI provide scientists like you access to, or free access to, world-class experts and state-of-the-art equipment to enhance your non-proprietary research beyond what your own laboratory could provide. Access to user facilities is determined through a competitive peer review proposal process. Now, because EMSL and JGI are funded through the same Department of Energy program, the Biological and Environmental Research Program, we have a common science mission, and that is to understand, model, and predict the biological processes underpinning environmental systems to enhance research protecting U.S. energy and environmental security. Now, you can find more detailed information. Sorry, I'm grabbing a link here for everyone. You can find more detailed information about this call on EMSL's website, emsl.pnnl.gov, and a link to this proposal call is in the chat window. Now, EMSL and JGI launched the FICUS Proposal Call in 2013 to further accelerate our users' research beyond what each of our facilities could contribute separately. Our collaborative and multidisciplinary approach to scientific inquiry continues to expand by introducing new expertise and resources available at other user facilities like CSMB and the NSF-funded user facility NEON, the National Ecological Observatory Network. Now, as a quick plug, the NEON-focused webinar is happening on March 4th. So for more detailed discussion on what NEON can offer you, I encourage you to join that webinar as well. And here is a link to that registration site as well in the chat window. So now I give the floor over to Scott Lee, our structural biology expert. Thank you.
Thank you, Linda. I'm going to go ahead and share my screen now. Are we good? Okay, so my name is Scott Lee. As Linda mentioned, I am Structural Biology Integrated Research Platform Lead, and I'm going to talk a little bit briefly about our structural biology capabilities here at EMSL. It falls into a number of categories. Up here in the upper left-hand side of the screen, it's our TEM capabilities. We have a new generation, a third generation Krios cryo-TEM that's been in operation for about 7 or 8 months now. That system can give you protein structure on proteins of about 100 kilodaltons or larger through single particle analysis. The instrument can also provide microelectrode diffraction, so you can get structural information on small molecules or even small proteins. And it also has the ability to do tomography, so you can get three-dimensional perspectives and relationships between subcellular organelles and whole cells. To support this, we have a cryo-FIB-SEM, focused ion beam SEM capability that allow us to prepare thin samples, lamella, that we can use for TEM, but in and of itself can also provide serial block face imaging for three-dimensional structure. In support of this, we have a cell-free expression pipeline for protein synthesis. We can, as long as we get the DNA, we're able to get proteins with enough sufficient purity to be able to run it on our TEM, but we can also do unpurified and do functional annotation as well. For proteins less than about 30 kilodaltons, 35 kilodaltons, we turn to solution NMR. And it's actually a very nice complement to cryo-TEM, and it provides access to those proteins that we couldn't do with cryo-TEM, but it also provides dynamics and information on intrinsic disorder as well. At a higher level, we can do native mass spectrometry, and that'll give you information about relationships between different subunits, stoichiometry, and ligand binding when you have proteins that are or protein complex with non-covalent interactions as well. And then I'm not going to go into detail today, but we can also do 3D image reconstruction using our software running on Tahoma. So this is our cryo-TEM. Like I mentioned before, it's been in operation about 7 or 8 months now. This is an example of the data that we have. It is 1.8 angstrom map of apoferritin collected on 3 days of automated data collection and processing. We like to point out the fact that the Krios, for the purposes of this call, is through EMSL access, but because the instrument was funded through a different source, if you only want to have access to the Krios, there is another opportunity for getting access to it. But if you want all the other capabilities associated with the BioSANS and the other capabilities I'm going to mention, you're going to have to go through the EMSL access as well. Here are the 4 different workflows that we are able to get with our system. Single particle imaging, here is where we are able to get protein structure at the 1.8 angstrom, 2 angstrom type of level as well. Microelectron diffraction will give us crystal structure of small molecules. For proteins that crystallize, we can actually get structure as well through micro-ED. And then finally, tomography is an example of what we can do to be able to get whole cell imaging in a 3-dimensional context. The Aquilous cryo-FIB-SEM basically has a number of capabilities. It can be used for serial block face imaging, like the figure in the upper left. And this is just a picture of bacteria interacting with a virus. But we can also use it to pull out samples from a specimen that we can use for TEM. And this is just to show you how we do this. The sample's in frozen water, which we put on a copper base, and then we can pull it out with a cryo probe. And then we do some thinning techniques with the FIB to get it in a state where we can do cryo electron tomography or micro-ED. The other thing we have is our protein pipeline, cell-free protein synthesis pipeline. And we have a robot on site that can actually generate proteins in 2 different formats. For cryo-EM structure information, there's a format called MAXI where we can generate 6 proteins and get the protein at 98% humidity purity in 2 days. But also a different type of method of getting this is through MIDI, where we're able to get proteins at a non-purified state, but we can get 24 proteins within 1 day, and that'll enable us to do functional annotation. For our structural biology for smaller molecules, we resort to NMR. This involves a number of processes. ranging from sample preparation through gene expression, protein expression, isotopic labeling, protein purification. That is something that you guys can do at your home institution, but we can also do that here as EMSL if needed to be. We collect the data with an NMR, go through chemical shift assignments, and then resort out to the solution structures in the upper right. So the nice thing about NMR is because it's in solution, you can get information on dynamics, disorder, and the binding surface. And this little schematic here on the left-hand side of the upper right shows the various binding, the black sort of domains within the protein that have a little bit of fluidity to them. So it shows you the dynamics that can occur within the proteins. And then finally, talk about mass spec. Normal mass spec capability. If you're working in a denaturing mass spec, you can get the protein complexes that are non-covalent, the subunits which are not covalently bound to each other to disassociate, and you can get the intact mass of these individual subunits, but that doesn't provide you information on sort of the stoichiometry or how these things are assembled. So what you need to do is actually put it in a different type of solution, and then through that, the protein complex does not dissociate, and that provides us information on the stoichiometry, how the subunits are bound to each other, what ligands are bound, what ligands or metals are bound to which of the subunits, and so forth. This involves an instrument with high mass to charge ratio, and it is something that we have here at EMSL, and we can do that work. So I'm going to stop here and sort of list the people that are sort of the boots on the ground, people who know the ins and outs of all the details of the individual capabilities for your reference. And so I'd like to turn it over at this point to Hugh O'Neill at Oak Ridge, who's going to talk about the BioSANS capabilities.
Okay, thank you, Scott. Okay.
Yeah, there you go.
Okay. Okay, so hello everyone. My name is Hugh O'Neill. I'm talking to you from Oak Ridge, Tennessee. I'm delighted to have this opportunity to tell you a little bit about our Center for Structural Molecular Biology, and hopefully give you some ideas on how you can incorporate our tools into some related work on a FICUS proposal. So at Oak Ridge, we have two neutron scattering neutron sources. We've got a high flux isotope reactor, which generates neutrons through a fission reaction. That's the high flux isotope reactor has been here at Oak Ridge and in operation for about 50 years. And more recently, we have a spallation neutron source. It generates neutrons by bombarding a mercury target with a proton beam. And that's a more recent facility and has been in operation since 2007. And between these two facilities, then we operate 30 beam lines. They're open. We're a national user facility, so they're open to users from the US and all over the world. And we, of course, support a wide variety of science across materials, chemistry, physics, and also biology. And of the 30 instruments between the two facilities, typically about 12 instruments are used for biology type experiments. So the Center for Structural Molecular Biology is unique among the resources at Oak Ridge, in that the other resources are funded by BES, and we're actually funded by BEOR. We operate the biological small angle neutron scattering instrument, and that's located at the high flux isotope reactor. It's circled here in red. And we also operate a biodeuration program in support of our scattering program. And I'll tell you a little bit more about that later. But now this picture gives you an idea of the size of a SANS instrument. The front tank is the BioSANS instrument located at Haifa, and then you can see the general purpose SANS, its sister SANS instrument behind it. So you can do a wide variety of biological studies using small angle neutron scattering. And I'm showing a few examples here. So on the top here is an example looking at biomolecular complexes in solution. So this particular example studied a flexible and intrinsically disordered protein and the ensemble of conformations it assumes in relation to when it's in a complex with other proteins. We have a large program in studying biomass, so we can extract the structural features of components of biomass, including cellulose and the matrix copolymers, and we can see how those structural features change under temperature and pressure when we break the biomass down to make biofuels. And Marcus Fostens is going to give us some insight into how he used these tools to look at for biofuels related work in his research. We also can study live cells, but neutrons are inherently non-destructive and don't cause any radiation damage. So that actually allows us to look at intracellular components under native conditions. This particular example was actually looking at a differentially labeled plasma membrane in a living cell. Of course, we also look at hierarchy assemblies such as membrane structures and how they interact with different proteins. So they're just a few examples of the types of measurements we can do using small angle neutron scattering. So what sets neutrons apart from other techniques? So X-rays, for instance, scatter off the electron cloud, so they're more sensitive to the heavier elements, whereas neutrons scatter off the nuclei, so you can actually see the lighter elements such as hydrogen with neutrons. And in addition, neutrons interact with hydrogen and its isotope deuterium very differently. So they're very different cross sections, and you can take advantage of that by varying the contrast in your solvent. So because different biomolecules have different hydrogen contents, you can actually separate the scattering contributions from protein, DNA, or protein and lipid in a complex by introducing deuterium during production of your biomolecule. So by deuterating a protein, we can actually then get more increased contrast between a deuterated biomolecule and other proteated components. So in the bottom left here is an example of studying a membrane protein. This is an aspartyl protease embedded in a lipid matrix. So one contrast, and that would be at near 100% D2O, because there's nothing actually deuterated here. We can see the protein lipid complex. And by going to the match point of a lipid, we can hide the contribution of the lipid and see only the structural contribution from the protein embedded in that matrix. So this slide gives you an idea of what's involved in doing a neutron scattering experiment. So I've mentioned some of the thematic science we do in terms of looking at biomacromolecules, membranes, hierarchical systems such as biomass. And we also can measure a wide variety of biomimetic and biohybrid systems. So in terms of sample types, we can measure solutions, so proteins in solutions, similar to what you would do in an NMR experiment. We can measure suspensions, so we can measure intact cells, or even very complex materials such as soils, and also solids such as plant cell walls, or even porous materials. If you're interested in looking at rocks, for instance, looking at the porosity or pore size distribution in rocks, we can do these types of systems as well. And in terms of sample requirements, so for solution scattering, which would be proteins and even membranes, we require about 0.3 to 0.5 mil solutions. For proteins, typically we would have about 2 to 5 mg per mil protein, so it's not a huge amount of material. And then for solids, depending on their density, we're talking about maybe 10 to 20 milligrams of solids. And these materials can be up to 1 centimeter in thickness, but generally we measure materials that are about 1 millimeter in thickness. So this is a schematic of the BioSANS instrument. The sample essentially sits in the beam in a cuvette or other measurement device. And the neutron scatter off the sample, we collect the signal on a two-dimensional detector, then we reduce that data, and then this is typically what a reduced curve looks like. So this is a SANS curve, and from that, we can extract information about size, shape, and aggregation or assembly of a biomaterial in a solution or pore size distribution of a solid. So lastly then, I briefly mention other capabilities we have. So because deuteration is so important for neutron scattering experiments, we have an in-house deuteration laboratory that's accessible to all our users. We have capabilities for fermentation for producing large amounts of deuterated materials. We also can do protein purification and characterization. And then, you know, we've developed protocols for doing various types of both HT labeling of proteins and other biomolecules, as well as producing deuterium labeled small molecules as well. So some of our support laboratories include biophysical characterization, where we have fluorescence, circular dichrosum, all these different techniques that you would typically find in a biochemistry laboratory. We do chemical synthesis to do produce small molecules. And we also have X-ray crystallography and scattering capabilities. So I'll leave you there today. I'll try and get this back up at the end. So if you have any more questions, you can contact me by email or visit our website. So our next presentation then is from Dr. Himadri Pakrazi from Washington University of St. Louis. And he's going to tell us about how he's used neutrons in his research to study cyanobacterial structure and dynamics. So Himadri, I'll hand it over to you now.
Thanks, Hugh. I'm going to talk about the organisms that we study in our lab. That's cyanobacteria. And I'm speaking to you from St. Louis, Missouri, from Washington University in St. Louis. So these cyanobacteria, as well as some members of my research group, over the last 15, 16 years, visited a number of the facilities that we are talking about. We have been fortunate to have close collaboration with EMSL and also with the BioSANS facility at Oak Ridge. Some of the organisms that we study, their genomes were sequenced at JGI, and there are other BER facilities where our research has taken us to them also. So cyanobacteria are oxygenic photosynthetic microbes, and as we all know, photosynthesis is the biological engine for solar energy-driven carbon capture and utilization. Photosynthesis in cyanobacteria, the same kind of photosynthesis which is oxygenic that we find in plants, they evolved first in cyanobacteria, and as a result of that, cyanobacteria have brought oxygen to the biosphere. Machinery for light capture and energy conversion resides in the thylakoid membranes. These are present in cyanobacteria as well as chloroplasts of plants. And here I show a cartoon diagram of such a membrane system, the thylakoid membrane, in which there are protein machines that are embedded. And the one that we will talk quite a bit about is this huge protein complex called phycobilisome that's attached to this photosystem II complex. So these proteins are either intrinsic membrane proteins or they are extrinsic proteins that are associated with membranes. We study these proteins in the context of cyanobacteria that are grown here in liquid culture, as well as on plates. They are simple bacteria, easy to grow, and we have been working with them for a long time. Now, this diagram shows you an electron microscopy tomographic reconstruction of a cyanobacterium And all I want to point out is that in green color here, pseudo color is shown that specific kind of membranes that internal to the outer membrane envelope of these gram-negative bacterium. Thylakoid membranes occupy about 15% of the cellular volume. Now, today we are going to discuss the antenna proteins. That's a special term. That's antenna for harvesting light. Imagine that those are antenna that are bringing in solar energy into the reaction centers or the business end of photosynthesis. And the one that we will talk about are phycobilisomes that are attached to the thylakoid membranes. Here is a cartoon picture of that phycobilisomes. The actual structures have recently been resolved using cryo-EM. These are very, very large protein complexes that are sitting on the top of the membrane as shown in this diagram. So what we wanted to find out is how are these phycobilisomes attached to membranes and how do they control the membrane structure? And in this case, work that was done at the BioSANS facility was extremely useful to us. Here I show 3 different mutants of a wild-type cyanobacterial strains. And basically these mutants do the following. Here is the structure of the phycobilisome. There are rods and there are cores. One mutant has only one phycocyanin hexamer. Phycocyanin is the pigment that's present there. The next one has the phycocyanin rods completely gone and has only the core of allophycocyanin, another pigment. And then this mutant PAL completely lacks assembled phycobilisomes. Now, as you see in this diagram, first of all, the color changes are interesting, but also from the point of view of the membranes, the amount of protein that's sitting on top of the membranes is quite different, as you can see here. And this is something that we wanted to look at on the SANS. And the question that we asked is, how do cyanobacterial thylakoid membranes respond to changing environmental conditions? This is from some time ago, and there is Hugh O'Neill along with other colleagues, and I will name them later on. Here are 2 members of my research group who visited that BioSANS facility. At that time, here were the cuvettes that were used. That's some time ago. And you can see the color differences between wild type, the CB, CK, and the PAL mutants. And these were put in this controlled environment. And the beauty of the SANS facility for us was that we can put live cells in there for a very long period of time, days in some cases. And neutrons, since they are not destructive, as you just heard, did not affect the photosynthetic physiology of these cells because we tested those after prolonged neutron scattering experiments. And what we found are these remarkable curves here. And we saw that the wild type CB, CK, and PAL, the intermembrane distances in live cells were as shown here, 62, 52, 45, and 38 nanometer. These matched quite nicely, but not exactly, with electron microscopy experiments that were done previously. And as you can see, that 62 corresponds to 55 and so on. These were done in live cells, and this was one of the pioneering experiments with this system that anybody has ever done. Another set of experiments that we did, this is with environmental effect on phycobilisomes, this antenna. If these cyanobacteria are deprived of a fixed nitrogen source, that results in phycobilisome depletion. This is something that we have known for quite some time. And we wanted to find out that in this reversible system, where you can deplete of nitrogen and sequentially over a period of 48 hours, you lose all phycobilisomes. But then what is not shown here is addition of nitrogen as shown here, where this thing reverses itself and phycobilisomes are naturally reproduced inside the same cells. The cell colors tell you that here there is phycobilisome, here there is none. And there again, we could follow the membrane dispositions inside these living cells by using SANS. And this was also published later on. I want to show you how the same system, the same cyanobacterial strains then visited the EMSL facility. And just to give you a very one-slide presentation of the remarkable work that was done on top-down proteomics of these intact phycobili proteins. Same system here. And what we looked at were the modifications in top-down proteomics of individual protein components of phycobilisomes. We were looking at these post-translational modifications, and what we saw is that over a period of time, when the system was going from with phycobilisome to no phycobilisome, first things that happened are tagging of these different proteins with specific kinds of post-translational modification. Here I show glutathione of specific residues of those proteins. This is again, this was the beginning of a completely new way of looking at this natural degradation and resynthesis process. So in summary, I will say the experiments using neutron technologies at Oak Ridge National Lab were designed to go beyond static images of cyanobacterial thylakoid membranes. SANS identified structural differences between phycobilisome and antenna truncation by gene deletion, as well as nutrient starvation. And then I want to point out the suite of technologies at EMSL have yielded comprehensive views of the structure and composition of cyanobacterial thylakoid membranes. I gave you only a snapshot in one slide. Over the period of last 16 years, we have published more than 50 papers in collaboration with researchers at EMSL, PNNL, and others in a collaborative environment. And I want to leave you with this statement: combination of the complementary technologies at the BioSANS and EMSL should provide a detailed systems level understanding of the energy producing systems in the biosphere. Here is my lab research group. This is before COVID. And our lab is funded by both BER and BES of the US Department of Energy, as well as National Science Foundation and the Gordon and Betty Moore Foundation. The people whose work that I mentioned, strong collaboration with Volker Arban, Brad Odell, Hugh Qian, and Hugh O'Neill at BioSANS. At EMSL PNNL, John Jacobs from Dick Smith's group and Dave Copinall have been close collaborators over a very long period of time. If you are interested in further information of what we have done, you can visit our website that's shown down here. Thank you for your attention, and I'll be happy to answer questions that you can put in the chat function. Our next presenter is Marcus Foston, my colleague at Washington University in St. Louis.
Okay, well, thank you. Madre and I are right down the hall from each other, but we both end up using a lot of EMSL and BioSANS resources. So this is, we end up on the same webinar. And so some of the work that my lab focuses on is more related to biomass conversion. So taking lignocellulosic components. And so if we're talking about lignocellulose, we're talking about woody, grassy plants that have carbohydrates, hemicellulose and cellulose, and then another polymer, lignin. And so our lab has focused on a wide range of kind of biomass conversion technologies and has worked with the BioSANS group at Oak Ridge to do neutron scattering actually on biomass itself as it's undergoing pretreatment. The idea is that the biomass needs to be degraded in some way so that then enzymes, typically cellulases, can come in, have access to the cellulose and break down the cellulose into sugars for fermentation. In understanding how the kind of structural reorganization of the biomass occurs has been an outstanding question in the field. And I think neutron scattering in particular can give unique information. And so there's been an ongoing collaboration between myself and the Hughes group for almost 10 years now, working on various components of that project. However, I'd like to tell you a slightly different story here, where we've been able to utilize both the facility at EMSL and at Oak Ridge in the BioSANS to do some really interesting work when we're looking at kind of intersection between more traditional chemical catalysis and biocatalysis. In this particular case, we're looking at integrated multi-omics analysis to the prediction of metabolic flux conversion, metabolic flux in lignin conversion. So in this particular case, we're taking the strategy. Lignin is this really recalcitrant material. It's difficult to break down. There are some enzymes that can start to break down lignin, but they chew the lignin up really slowly. And so the question is, can we speed that process up, but then still take advantage of the real rich metabolic pathways that already exist in nature to metabolize aromatic species that come from lignin? Lignin's everywhere in every plant, right? So there's a ton of natural ways that microorganisms take advantage of this. Then can we then redirect that flux towards really useful flux and chemicals? So our approach has been this hybrid approach where we apply chemical methods, catalytic methods to make what we call lignin breakdown products, and then take those lignin breakdown products and feed them to a microorganism that can funnel, that has various pathways that can funnel the heterogeneous mixture of compounds that we get out of lignin towards a valuable product. And we do this for two reasons. Maybe you don't want aromatics as a product, or maybe we can never make those aromatics selectively enough so that they're amenable to separations. So we're using this as a funneling mechanism to increase our selectivity towards a product. In this case, we've been relying a lot on a Rhodococcus opacus strain. And in this particular research, we were really interested in interrogation of the metabolic pathways and genetic regulation to control utilization and tolerance of the lignin-derived kind of aqueous soluble phenolics that we might get from a chemical breakdown process, such that these Rhodococcus microorganisms could consume those and produce a product, in this case, triacylglycerides, which is a biodiesel. Where we started was we did some adaptive evolution experiments where we evolved mutants that could tolerate and utilize carbon sources, things like phenol and glycol, hydroxybenzoate, things that may be in a lignin breakdown product. Through directed evolution, we produced these mutants that were able to tolerate them at higher levels, and then also consume them at greater rates. And what we found was that a lot of the tolerance and utilization came from upregulation of various pathways. We didn't see a lot of actual mutations, genetic mutations, and this was mainly controlled through upregulation. So then the idea was like, well, if there is a specific transcriptome that can give us the kind of flux that we want, is there a way that we can do this and predict this beforehand? So we started working with Hector Garcia Martin at JBay at Berkeley to try to enhance genome scale models to specify a transcriptic solution space that could give us a desired metabolic output. To do this, though, we needed a large number of transcriptomes to do this, and then we need to be able to map that onto real fluxes. So that's where some of the expertise of EMSL and JGI can come in. They have these really nice pipelines that are designed to do large numbers of analysis such as this. And so we sent, for example, a large number of samples using the kind of integrated research platforms in both the biomolecular pathways and cell signaling communication groups at EMSL, where we were then able to send not only a bunch of data for get a lot of proteomes and transcriptomes developed to kind of build this omics to flux models, but we were actually able to do this for supplement this with some metabolome data. So what I'm showing here on the left is a qualitative analysis of our wild type rhodococcus grown on different substrates. And we're able to kind of track the metabolite pool concentrations normalized by biomass. And what we see is that we can actually then measure the kind of flux that are going through these systems. Using this information, in theory, we should then be able then to constrain the rhodococcus genome scale model and come up with a way of then predicting flux based off of a transcriptome, and in this case, off of a metabolome profile. Just to kind of switch gears, though, to understand what we're feeding the rhodococcus, we need to break down the lignin. And so this is where we use our neutron scattering. And so this is a neutron scattering. This is a BioSANS line. And what we've done is we've developed, Hughes developed a high temperature, high pressure cell that we can put inside of the neutron beam, and we can actually see the lignin breaking down as a function of time. So what we've done is we're trying to understand how lignin breaks down so that we can understand what we're feeding our rhodococcus. And from this, we're actually able to gather a range of small angle neutron scattering profiles. These profiles tell us something about the size of the lignin that's in our solvent as we're breaking it down. And so we were able to extract information like this, where this is basically a size distribution for our lignin in our reaction mixture at 250 degrees as a function of time. And we can see here that the size distribution of our lignins are changing as a function of time, and the amount of those in solution, or our volume distribution, is changing as a function of time. Taking advantage of some of the unique capabilities of neutrons, not only because they don't have a lot of damage. We can do this on organic materials. It's sensitive to organic materials. And also we can do these contrast matching experiments, where now we remove the contribution to our neutron scattering profile from a third component, a third phase, which is our catalyst in this case. And so now we're able finally to understand kind of like the size of our lignin in solution and the volume distribution, but analyze that as a function of with and without catalyst. Again, kind of understanding how the lignin is falling apart in the solvent as a function of time. But as you notice, neutrons tell us nothing about the chemistry itself. And so that brings us back to our EMSL resources, where a lot of magnetic resonance and mass spectroscopy tools typically devoted to things like soil organic matter could also be applied to our lignin as it's breaking down. This is where we use high resolution mass spectroscopy to try to understand, again, the complexity of the chemistry that's changing. Here we've also used high temperature NMR to also correspond with some of the things that we've measured in the BioSANS at high temperature as well. These high temperature, high pressure NMR setups were actually really developed for biogeochemical transformations. So you can imagine a mineral carbonation reaction mechanisms being inserted here, or altering of subsurface microbial biochemical behavior in the presence of supercritical CO2. But we did this as a function of lignin depolymerization. And this is kind of some of the data that we can get out, where we're able to look at chemical changes occurring to, in this case, a model polymer of lignin as a function of reaction time. I didn't have a really good conclusion slide here, but it's a number of people that went to bat for us here that did a lot of work, both at Oak Ridge and the BioSANS line and at EMSL. And I had a lot of great graduate students and people and groups funding us as well. And so with that, I'll end my presentation.
Of course, I was on mute. Sorry about that. Well, thank you for all of your great presentations today. We didn't have a whole lot of questions in the chat window, so I guess the presenters were very thorough. in their explanation of how they were using BioSANS. But what we can talk about just at this moment is reiteration. For this particular FICUS call, the letter of intent is due on March 17th. So if you're planning on submitting a proposal, make sure that you get in a letter of intent first by March 17th so that our staff have a chance to review your proposal project and get you feedback on it. Full proposals are due on May 3rd. And, you know, this is your time. We've got another 15 minutes left. So if you have a burning question, feel free to stick it in the chat box. Do any of the presenters have any other information that you want to make sure our audience hears before we close out this webinar today? Anything that you want to emphasize again?
Yeah, I don't have any specific information. I think you've covered everything about the call, but if you have any questions, you should feel free to reach out to me. I can stick back up my information just for a minute, just so you,
All right.
yeah. So you feel free either to email CSMB, the general email box, and that's pretty easy to remember, or me directly, and I'll get back to you. So yeah, any questions you might have about how you can apply our techniques, I'd be more than happy to discuss with you offline.
Wonderful. Hey, why don't you copy paste that URL and put it in our chat window so that
yeah,
access that. And then also I'm going to re-input the link, the direct link to the proposal page on the EMSL website where you can read more about this FICUS call and then you know, have a link right there to submit your letter of intent. So actually, I did not send it to the right thing. All right, and there, sorry. All right, and Hugh, if you can do the same, that would be great. This webinar was recorded. For those of you who may not have noticed that, we are going to take this recording and put it on YouTube as quick as we can. And then based on our registration site, we've got your email. So I'll send an email out to everyone with a link on YouTube to where you can access this recording. All right, great. Well, it sounds like nobody's got any other questions. So have a great rest of your day. We hope to see a lot of letters of intent from everyone. So have a great day. Thanks, everyone.
Thank you very much, everyone.